Cancer Nanomedicine. Opening. Cancer Nanomedicine aims to overcome key limitations in conventional cancer diagnostics and therapies. Despite impressive academic output, translation to routine clinical care has been uneven, raising questions about impact and maturity. In this viewpoint, four leaders offer complementary perspectives on what nanomedicine has achieved and what it still needs. In this video, I will summarize their answers to 5 practical questions: approvals versus publications, opportunities in detection and diagnosis, how to improve delivery, advantages over conventional drugs, and how to strengthen preclinical testing. The disconnect between research and approvals. First, why do so many nanoparticle papers exist while relatively few products have FDA approval? Sangeeta Bhatia argues the question is misleading. We don't judge modality by immediate oncology approvals in a single disease. Historically, modalities like monoclonal antibodies, siRNA, gene therapy, and CRISPR matured over decades and often found their first approvals outside oncology. The COVID-19 vaccines are a case in point. Long-term investment in lipid nanoparticles enabled rapid, safe deployment of mRNA at scale. Today, nanomedicine extends well beyond smart bullets for chemotherapy. It now synergizes with immune checkpoint blockade, adaptive cell therapy, cancer vaccines, and surgical margin detection. Xiaoyuan Chen highlights 3 scientific and practical bottlenecks: unclear toxicities and nanovalue, especially nanoimmune interactions, limited reproducibility, and transparency in synthesizing precisely tuned particles, and complex, costly CMC GMP manufacturing with evolving regulatory expectations. Marina Dobrovolskaia frames that disconnect as a classic innovation trajectory: early hype, disappointment, and a long maturation curve. Liposomal drugs illustrate this. Periods of enthusiasm and retrenchment preceded viable products and a substantial market. Similar arcs occurred for siRNA and mRNA, technologies that ultimately relied on nanocarriers to succeed. Translation is slowed by high cost, investor uncertainty, regulatory gaps, and public skepticism, the valley of death. Twan Lammers adds a corrective: more than 15 cancer nanomedicines are approved, comparable to antibody-drug conjugates. A key reason for a slow perceived impact is patient stratification. For biologics, biomarkers are routine. For nanomedicines, especially non-ligand targeted formulations, stratification is harder. Imaging-based selections, for example, PET SPECT to gauge tumor accumulation, helps, but is not yet pragmatic at scale. Can nanotechnology advance detection and diagnosis? Early detection must identify lethal cancers earlier while minimizing false positives. Bhatia notes nanotechnology can amplify weak signals, multiplex analytes, and pair with AI to improve sensitivity and specificity on top of complex human biological noise. Importantly, several high-value analytes are themselves nanoscale circulating tumor DNA in nucleosomes, extracellular vesicles, and engineer synthetic biomarkers that release nanoscale reporters. This positions nanotechnology to power next-generation diagnostics, including in low and middle-income settings where leapfrogging heavy infrastructure may be possible. Chen distinguishes in vitro and in vivo applications. In vitro, Nanoparticles boost surface area and enable multiplexed, highly sensitive detection using platforms like quantum dots, gold nanoparticles, and polymer dots. In vivo, radio-labeled nanoparticles can map sentinel nodes. Iron oxides supports MRI, and micro-nanobubbles enhance ultrasound. Multimodality probes become feasible by integrating functions into a single particle. Dobrovolskaia cautions that, despite femtogram-level sensitivity, the lack of validated clinical biomarkers limits widespread adoption of nano-enabled diagnostics. Lammers is skeptical about 20 to 200 nanometers multifunctional probes for general tumor imaging, noting competition from established PET, CT, MRI tracers and the viability of nanoparticle tumor uptake. However, he acknowledges clear niches: intraoperative tumor margin visualization, sentinel lymph node mapping, and detection of small nodal metastases. How do we improve nanoparticle delivery? Delivery is a multi-step journey: circulation, extravasation, interstitial transport, cellular binding, internalization, cargo release, and action. Bhatia points to deeper biological design: co-opting serum apolipoproteins for liver targeting, albumin hitchhiking to reach lymph nodes, and virus-inspired transport peptides to penetrate tumor tissue. In parallel, unbiased high-throughput strategies like barcoded libraries systematically discover delivery rules and winning formulations. Chen argues the field over-relied on enhanced permeability and retention effect, which is robust in xenografts but inconsistent clinically. He advocates smart particles that transform size, charge, or shape by internal cues, like pH or enzymes, or even external triggers like light, heat, or magnetism, and designs that target stromal and immune cells in the tumor microenvironment, or even lymphoid and myeloid compartments, expanding beyond tumor cells alone. Dobrovolskaia emphasizes linking physicochemical features to biological outcomes for each drug carrier pair. Success with one API rarely generalizes. She highlights the need for predictive algorithms and attention to biological rhythms and sex hormone cycles that may modulate nanoparticle pharmacokinetics. Lammer reminds us that delivery often fails due to tumor physiology, poor perfusion, dense stroma. Priming strategies like vascular normalization, stromal remodeling, and local co-treatments like hypothermia, radiotherapy, or ultrasound can help, but systemic metastasis limit strictly local methods. Also, active targeting typically fine-tunes cellular uptake, but bulk tumor accumulation. It's crucial for cargoes like siRNA that must enter cancer cells, less so for small molecules that can be released and redistributed within the tumor. Advantages of nanotherapeutics over conventional drugs. Nanotherapeutics can reshape pharmacokinetics and biodistribution. Longer circulation, smaller volume of distribution, and reduce off-target exposure. They can stabilize labile or immunogenic cargoes, for example mRNA, and co-formulate multiple drugs at fixed ratios, useful for combination therapy. Clinically meaningful toxicity reductions are already evident. Albumin-bound paclitaxel avoids chemophore-related hypersensitivity. Liposomal doxorubicin reduces cardiotoxicity versus free doxorubicin. Beyond delivery, nanoscale physics enable modalities like magnetic hyperthermia and radiosensitization, and support immunotherapy by targeting antigen-presenting or myeloid cells, or serving as in vivo depots and artificial antigen-presenting platforms. Strengthening preclinical testing. Bhatia advocates multi-institutional ecosystems that share data, assays, and translational know-how. Chen recommends more predictive models, patient-derived xenografts, humanized mice, and phase zero microdosing with labeled nanoformulations to de-risk pharmacokinetics and biodistribution before dose escalation trials. Dobrovolskaia proposed the 3A framework: mechanisms of toxicity for API and carrier, markers that capture immune variability and predict liabilities, and models that better mirror human immunity, including naturalized mice or, when appropriate, large animals for infusion reactions. Lammers calls for standardization, head-to-head comparisons with standard of care in agreed-upon tumor panels, and critically publications of failures to close knowledge gaps and accelerate progress. Biomarker development for patient selection should start preclinically, as it has for most other oncology drugs. Conclusion: Cancer nanomedicine is not a failed promise, but a mature modality with proven products and expanded roles in therapy, immuno-oncology, imaging guidance, and diagnostics. The path forward hinges on four levers: robust biomarkers for stratification, biologically informed and high-throughput delivery design, clinically predictive preclinical models with transparent reporting, and scalable, standardized manufacturing. With these, nanomedicine can deliver earlier detection, safer treatments, and more durable outcomes for patients with cancer.